Okay, so today it's cartilage and bone, and these are skeletal connective tissues. They are analogous to the connective tissues we talked about the other day, except that they have an extracellular matrix that makes them more rigid, and they can support weight and do other things that the other connective tissues couldn't. So I want to start with this picture, the skeleton, and everything that's shown in white is bone, and everything that's shown in this dark pink is cartilage. And this is in the adult body. And as you'll see later on in the term when I talk about the development of cartilage and bone, most of these bones, particularly the long bones, have come from cartilage templates in the embryo and fetus, and the bone has replaced the cartilage. So the initial development of most of the bones is by chondrogenesis, or cartilage formation, and bone is secondary to that. But in the adult, we have mostly bone in the skeleton. The anterior portions of the ribs are cartilage. Cartilage is flexible where bone is not. And if we fill our lungs with air, we want our chest cavities to be able to expand and readjust. And cartilage is suited for that. If the rib cage were just solid bone, there wouldn't be much capacity to expand. Another place that cartilage is extremely important is on the articular surfaces. It's because if you had bone just rubbing against bone, it would be a high friction surface, and cartilage is slippery. So it can bear compression and spring back, and it slides past other surfaces of cartilage. So that's very suitable as well. The nose and the external ear are also cartilage, and the intervertebral discs, which you don't want simply bone crashing against bone every time you walk or jump, and there's something kind of compressible and kind of restoring in cartilage. The rings around the trachea would normally be a floppy tube, and it has this external skeleton that keeps it open or patent, and those are made of cartilage as well. And so I'm going to talk about cartilage first. I'll talk about the cell biology, the extracellular matrix, and I'll point out that there are three main kinds of cartilage. But the major type is called hyaline cartilage, which means glassy cartilage. So first, before we get to that, let's just look a little closer at the rib cage. And I just want to point out that when cartilage meets bone, it's not gradual. There's a sharp demarcation point. So these are two very distinct types of tissue. And in the intervertebral discs, you could see that cartilage is interspersed with bone, but again, it doesn't grade into each other. They're very distinct. There are some tissues which kind of grade into other tissues, and you'll see when tendons meet bones, they meet bones at the portion of the bone that is surfaced with cartilage. And the tendon, as dense regular connective tissue, grades into the cartilage by a tissue called fibrocartilage. But we'll see that later. But this hyaline cartilage meets bone in a very abrupt fashion, and this fibrocartilage of the intervertebral disc similarly meets bone very abruptly. Okay, so here is a section of hyaline cartilage. And the cells are called chondrocytes. So the prefix chond, C-H-O-N-D, refers to cartilage. And as I mentioned when I talked about extracellular matrix, a very prominent molecule in cartilage is chondroitin sulfate, the glycosaminoglycan. And that is very abundant in cartilage. It was discovered in cartilage, but it's not exclusive to cartilage. But in general, every other thing with that prefix is exclusive to cartilage. So you have these cells, and they're large and bean-shaped. You can see the nuclei. The cytoplasm is stained pale here. And the matrix is this glassy matrix. In the living state, light will shine through it. It looks glassy. And one very striking thing about this is cartilage is avascular. Blood vessels are kept from growing into it. And in fact, if blood vessels grow into cartilage, the cartilage is either ready to be taken away as in development that happens, or it could be pathological. But you don't want blood vessels to grow into cartilage. But there are blood vessels in the surrounding tissue, which is called perichondrion, which is a dense, irregular connective tissue, which is vascularized. It consists of fibroblasts and type 1 collagen is the most prominent protein. But the inner region of the perichondrion, the part that is closest to the cartilage, is chondrogenic. It can give rise to new chondrocytes. And this is a process of differentiation. It's called appositional growth of cartilage. It means growth from the sides, from the edges. And it's a differentiation process because you have these cells that are not chondrocytes becoming chondrocytes. First they become chondroblasts, and then they become full chondrocytes. Okay. So the chondrocytes are surrounded by a rim of matrix that might stain a little differently. It used to be thought to be a different structure back in the 19th century before they knew much about cells and everything and matrix, but and they called it the capsule. And it's a term that's sometimes still used. Sometimes it's called territorial matrix, but it's just not fully hydrated yet. The composition is a little different because it's new, so it looks like a different structure, but it's really just part of the cartilage matrix. Okay. Now, cartilage is unusual as well, is that it can keep on dividing after it's differentiated. Many cells don't. Muscle cells don't, and nerve cells don't, but cartilage cells can. But they don't divide much. And when they divide, the cells don't move very far past each other, typically. So you have clusters of cells where you can tell they originated from the same progenitor. So these are called isogenous groups of cells. So here's an isogenous group. There's another isogenous group. They're just made up of chondrocytes that have a common progenitor chondrocyte. Sometimes you have clusters of 4. And sometimes they're not all in the plane of section, so you'll see 3 of them. But, and then sometimes you'll see cells that have moved apart from each other because more matrix has interposed between them, and so you're not sure whether it's an isogenous group or not. But the clusters are called isogenous groups. And the kind of growth that does this is called interstitial growth as opposed to appositional growth. Interstitial means growth from within. And it's important to realize that this is not differentiation. This is just replication of the cell, because the cell is already terminally differentiated and it's just dividing. So one is a differentiation process and one is just a replication process. Okay, here is kind of a lightly fixed tissue. The cells have shrunken in where they sit. And you can see that the matrix is quite stiff. It's very glassy looking. It's avascular. But the cells have shrunken. And because the matrix is so stiff, the matrix doesn't kind of close in on the shrunken cells. It just remains kind of rigid. And these little spaces that are exposed when that happens are called lacunae. Lacunae is just another term for space. And so the cells are sitting in the lacunae, but most of the time you wouldn't know it because they fill the lacunae. When they're alive and functioning, they're filling the lacunae. When they are dying and they're shrinking, you can see the space. And then you can see the territorial matrix or the capsule around the lacuna. Okay? And no blood vessels. Sometimes an empty lacuna. Okay? Now here's a schematic, and this is kind of showing us a little more detail. And the details have been derived from electron microscopy. So in the light micrograph, the cells kind of look smooth, but here we can see they're scalloped. They're kind of pointy. They have these points around them. And you'll see an EM in a few minutes that'll indicate that. The cells, the nuclei have nucleoli. They have rough endoplasmic reticulum. They have Golgi apparatus because they're secreting a lot of matrix. Here the territorial matrix looks relatively vacant. It doesn't have all the fibers that the interstitial matrix does. So that's a characteristic of when it's newly synthesized, the proteoglycan may get synthesized or secreted earlier, and then the fibers build up. Okay, and the collagen fibers, the collagen of cartilage is not the typical connective tissue collagen, type 1. It's type 2 collagen. Type 2 collagen is also triple helical. It also forms fibrils and fibers. But in the case of these triple helices, all of the 3 subunits are the same. They're all from the same gene. So there's just one type 2 collagen gene. And you can see the perichondrial fibroblast and perichondrion and appositional growth. Okay, so here is an actual EM, transmission EM. You can see the cells are scalloped. You can see the large nuclei. You can see the nucleolus, chromatin, the clumpy chromatin. You could see the rough endoplasmic reticulum and the Golgi apparatus. And in the matrix, it doesn't look glassy under the EM because things have been stained. So you see the collagen fibers. They're very thin. They're much thinner than the fibers of type 1 collagen. And there's a lot of granules, and these granules are collapsed proteoglycans and proteoglycan aggregates. So what are those? So you can do an EM like this where you make a section and you cut the section very thin and you stain the section. There are other kinds of EMs, as you've learned in previous lectures and in the lab. But one type that you may not be familiar with is that you can actually extract the material in the tissue, and you can just spread it on a surface, and you can visualize what was in the matrix when these large molecules spread out. So let me show you a schematic. And here in the cartilage matrix, we have collagen, and we have the proteoglycans that you've seen before. The central filament of these proteoglycans is the glycosaminoglycan hyaluronan. And that's here. Okay. Here, the color coding is not very good here, but actually this and this are proteins. And the gags, this arrow should be pointing to these little chains. The gags are little fibers coming off. So I'll give you a better picture of this in a minute. But what I want to show you here, forget about the colors and everything, is that you have proteoglycans that are stuck, arrayed along hyaluronan chain. And together this makes what's called a proteoglycan aggregate. And these aggregates are not that common. They're present in cartilage, and they're present in the stroma of the endometrium. But most connective tissues have proteoglycans that are not aggregated. So what does it mean that they're aggregated? So we're looking at this electron micrograph, and we see these little chains here, and these are all proteoglycans. They've kind of come off the aggregate. And then you have this long filament here with a lot of chains stuck to it. So what that really is, this thing, is that the central blue thing is hyaluronan. It's just a very long glycosaminoglycan chain. And then the green is protein of the proteoglycan. The proteoglycan is called Aggrecan, and the red sticks coming off it are the glycosaminoglycan chains. Two kinds, chondroitin sulfate and keratan sulfate. And they are aggregated along the hyaluronan by things called link proteins, which are these black balls here. And this linkage is a non-covalent linkage. It's called a hydrophobic linkage. So you can, if you apply a solvent, you can make it fall apart. If it's covalent, then it doesn't fall apart with solvents. Okay? So that's different kinds of molecular interactions. But what the effect is to make this very kind of structured, space-filling, huge molecular gamish, collagen, proteoglycans, proteoglycan aggregates, and the scale of this is more or less accurate. So this is the size of a cell, and these molecular structures are even larger than the cell. And there's a lot of water there. So it's filling a lot of space with these molecules. And that, that gives the cartilage matrix its particular physical properties. And one important thing about this is that ions and small molecules are permeable, or can permeate through this matrix. So if the matrix is not vascular, if the tissue is not vascular, how does it get nourished? It gets nourished from the edges, from the perichondrion, or sometimes it's attached directly to bone, and it gets nourished from those blood vessels, but then there's diffusion through the matrix. And if you've kind of felt a piece of cartilage, like the keel bone of a chicken, for example, it's a very dense tissue, but on a molecular level, it's also very permeable, and nutrients could get in and out, and small proteins can get in and out, and ions. So it can be nourished, and it can get rid of waste materials by diffusion. Okay, so this is a rabbit, and this is the same rabbit in all four pictures, and this rabbit lived in the 1950s. I don't know what it's doing now, but it was a, but anyway, this is a very famous experiment done by a famous research physician and writer, Lewis Thomas, and it told us a lot about cartilage, because rabbits' ears are made of hyaline cartilage. Our ears are elastic cartilage, which you'll see in a minute. This is hyaline cartilage, and they're very stiff, rigid, and the rabbit was injected with papain. And papain is an enzyme that is extracted from papayas. It's a proteolytic enzyme, and it breaks down proteins. And when the rabbit was injected with papain, the ears collapsed. So the papain went through the bloodstream, it got to the perichondrion of the ear, and it evidently got into the matrix, and it broke down proteins, type 2 collagen, and the protein of the proteoglycan, and the ears flopped. So what does this tell us? It tells us that the cartilage matrix is permeable, even to small proteins. It tells us that the cartilage matrix, the physical integrity, depends on the proteins, among other things. It may, you may have gotten the same effect if you broke down the glycosaminoglycans, but that wasn't this experiment. But anyway, breaking down the proteins makes the ears flop. So the proteins are very important for the structure. Yes? The cartilage in other areas? Probably it got softer. Yeah, it wasn't so evident because it wasn't so external. But it wasn't fatal. That was the important thing. The cartilage could bend over. Okay, so then the papain wasn't injected anymore, and the rabbit was allowed to recover, and the ears got stiffer, and ultimately the ears got back to normal again. So what does this tell us about cartilage? That cartilage matrix can be restored, and so that the biosynthesis of the chondrocytes is sufficient to put the matrix back. It's not irrevocably damaged, and it just came back to normal. The rabbit kind of survived the experiment. I mean, definitely survived the experiment, and so it kind of taught us a lot about the nature of cartilage, which a lot of this wasn't known at the time, actually. So now one important thing about cartilage, even though the matrix is repairable and the cells are biosynthetically active, the cells are not dividing very much, as I showed you before, and also cartilage cannot repair breaks. So if a cartilage is torn, like the cartilage in the knee, or if it's severed, it doesn't reseal the break readily. I mean, there are things that are being done now to kind of encourage more cartilage to grow and factors and stuff. But because it's not vascular, it just doesn't repair well. Okay, so now I'm going to talk about not hyaline cartilage, but fibrocartilage. So fibrocartilage is in the intervertebral discs, and it's in the pubic symphysis. And because it's directly attached to bone, it gets its vascular supply from the bone on its edges, and it doesn't have a perichondrion. Okay, so fibrocartilage doesn't have a perichondrion. And here is a section of the vertebral column, and this is bone here. And as I'll talk about later, it's not easy to cut a section with bone unless you take all the mineral out. So this had to be demineralized. But the bone is here. This is called spongy bone. There's normally marrow here, more spongy bone. These are vertebrae. But in between is an intervertebral disc. But the center of the intervertebral disc is very rich in proteoglycan and soft, and it tends to pop out during the preparation. So all you're left with in the fibrocartilage are the remnants around the side, remaining fibrocartilage. The whole thing is fibrocartilage, but the composition is different from the inside to the out. So you have the more fibrous part of the fibrocartilage over here. And no perichondrion. It's directly attached to bone. So this is a section And you must know by now that when you see this blue-green or turquoise color, you're looking at usually type 1 collagen in dense connective tissue. And fibrocartilage is like a dense connective tissue, staining blue-green. Ordinary cartilage, hyaline cartilage, won't stain like this. So what's going on here? It has a lot of type 1 collagen in addition to the type 2 collagen. So it has all the ingredients of hyaline cartilage plus type 1 collagen. So if you look at this whole thing, if I just showed you this little portion over here, remember Dr. Luria's picture with the girl in the waves, and you look over here and you just think, well, that's just connective tissue. That's dense, regular or irregular connective tissue. But then you kind of widen the scope. You look at low magnification and then you home in. And these are not connective tissue cells of the ordinary kind. These are chondrocytes. This is an isogenous group of chondrocytes. So when you see something that has the characteristics of dense connective tissue, but it also has chondrocytes, then it's fibrocartilage. Okay, so now in the larynx and trachea, we have cartilage rings and plates that keep the tube from collapsing. Okay. We also have a tissue here called the epiglottis, which is cartilage, and it's still a different kind of cartilage. This is elastic cartilage. We don't have too much of it in the body, but the epiglottis, the flap of tissue that prevents food from going down the trachea, is made of elastic cartilage, and our external ears are made of elastic cartilage. So elastic cartilage can be specially stained with a stain for elastin. I showed you examples of special staining for elastin when we were looking at elastic connective tissues. This is elastic cartilage. So you see there are chondrocytes, there are isogenous groups. There's a perichondrion with no elastin in it, but there's elastin in the interstitial matrix, and it tends to build up in the capsule region, but it's all in fibers and sheets throughout the matrix. And as the cells differentiate from the perichondrion, they become elastic chondrocytes, and they produce all the ingredients of cartilage, type 2 collagen, aggrecan, but also they produce elastin. And here's another view, elastic cartilage, low magnification, high magnification. This is specially stained. Sometimes in the lab you'll see a section that has elastic cartilage in it, but it's not been specially stained. But the elastic cartilage has this kind of uneven mottled appearance because it takes up the H&E stain differently than hyaline cartilage. But you can see the isogenous groups, and you can see the elastic fibers. The perichondrion stained differently. Okay, so now we're going to get to bone. And in order, yes?
So the chondrocytes in the elastic fibers, or sorry, the elastic collagen, they create both the elastin and the collagen?
Yes, the chondrocytes just have this capacity to produce elastin as well as everything else. You don't have separate cells doing that. Yes? So the question is, do they make type 1 or type 2? They make type 2 because only fibrocartilage makes type 1. You don't have any type 1 collagen in anything but anything but fibrocartilage. Yes?
So the primary way to distinguish between the elastic cartilage
The modeled appearance, yeah, I mean, from our point of view, if we're going to ask you to identify elastic cartilage, we're not going to show you some unevenly stained tissue. We'll show you a specially stained tissue. Okay. Yes? Yes, there's going to be fibrillin also, yes. Okay, so we're going to look at the middle ear, because the middle ear has some interesting bones called ossicles. The smallest bones in the body, the incus, the stapes, and the malleus. They're involved in hearing. And this is bone of the cranium. This is spongy bone, but these are bones too. And we're going to concentrate on these bones because I want to show you the relation between bone and cartilage at a joint surface. And so here is a good picture of this. This is a joint of an ossicle. And this is bone, and this is bone, and this is cartilage here. And one important thing is that two bones are coming together. They don't come together as bone against bone, but they come together as cartilage against cartilage because the articular surface in both cases is covered with hyaline cartilage. The hyaline cartilage is nourished by the bone. It does not have a perichondrion either at this side, and it doesn't have it here either. This is a specialization, because if it had a perichondrion, it would defeat the effect of the low friction surface. It would be high friction with perichondrion. So there's a fluid here. The fluid is called synovial fluid, and it's rich in hyaluronan, and it also has nutrients in it. Okay. So these are chondrocytes here, and the cells of the bone are called osteocytes, and there are blood vessels in the bone. And where the cartilage meets the bone, it's a very abrupt boundary. It's not a straight boundary. It's a very convoluted and contorted boundary, but it is abrupt. There's no question that it's cartilage on this side and bone on that side. And because of the resemblance to what happens like on a beach where the water washes up on the dry sand and there's no question about which side is wet and which side is dry, they call this the tide mark. The tide mark. It's where the cartilage meets the bone. Okay, here's another old experiment, very interesting experiment. This is the surface of a chicken egg, and it is a very rich source of vascularity. It's actually, this system has been brought back. This was back in the early 1960s, I think this experiment was in. But the system was revived. It's used in cancer research now to put pieces of tumor on the top of the chicken egg membrane and look at how it gets vascularized and how you can prevent it from getting vascularized. But this was just a demonstration about the properties of cartilage and bone. So this is a rib, and remember the rib had that abrupt boundary between the cartilage and the bone. And this is just a plastic ring, and it's holding down the piece of rib here. And it's on top of the egg membrane. And blood vessels are actually growing into this tissue. They're growing in to the bone side, but they're not growing into the cartilage side. So there's something in the cartilage that prevents the blood vessels from growing in. Okay, so the people back then said, well, is it something that the cartilage has that keeps the blood vessels out, or is it something that the bone has that makes the blood vessels come in? So they did a very simple experiment. They took a similar piece of rib and they just soaked it in a strong salt solution that would take out loosely bound proteins and molecules. And what they found then is that the blood vessels grew into both the bone and the cartilage. So that meant that the cartilage had something in it that prevented the vessels from growing in, that could be easily extracted with the salt solution. And these are called anti-angiogenic factors. And they're actually now used in cancer research because if they could be administered, possibly by gene therapy or just by putting in some kind of polymer that releases them at the site, and you could get tumors to have anti-angiogenic factors within them, they will keep the blood vessels from growing in and the tumors will die. Cartilage doesn't die, it's because it has this permeable matrix, but tumors would die. And it's working to some extent. It's being worked on. Okay, so I want to show you something about the structure of bone. And we can go in either direction here. Let's start with the bone. And we'll look at a piece of bone that's about 100 microns across. And we're looking at cells and the surrounding matrix. The matrix, the cells are osteoblasts and osteocytes. The matrix is type 1 collagen. And there's also proteoglycans and there's mineral. Okay, and we'll see how that gets there. The bone marrow stem cells can give rise to bone cells. So that's the source of them. Okay. Then if we go at a higher magnification, look more closely, we find that along these collagen fibers, there are very regular arrays of crystals. The crystals are called hydroxyapatite, sometimes with an L, hydroxyapatite, same thing. And they contain calcium, sodium, phosphate, water, and they crystallize. And because the collagen is itself paracrystalline, it has those regular epitopes along it, the mineral gets deposited in a very regular fashion. Then if we look very much closer, just at the collagen, we find there's a collagen triple helix. And there's this type 1 collagen in bone. So we go this way, the collagen gets studded with crystals. It is part of the matrix where there are cells that are producing it, and they make part of a bone. Okay, so what does the mineral and the organic material contribute to the integrity of the bone? So if you had a bone and you simply soaked it in hydrochloric acid, it would leach out all of the mineral crystals. No more hydroxyapatite. But the bone would not change its size or external appearance. Its substructure would be different, of course, but it would look the same. But you could take that bone and you could tie it up. It would be so flexible. So the organic matrix of bone is sufficient to keep its shape, to keep its size, but and its flexibility, but not its ability to bear weight. If we tried to stand on demineralized bones, our legs would bend. It wouldn't be good. So, so the bone remains resilient but loses its rigidity. If we instead left the mineral in place but removed the organic components with organic solvents, again, the bone would look the same, but it would have other properties. It would be very easy to shatter. I didn't have a shattered bone, but here's a pillar, and it shatters like that. And we know that our bones don't shatter very easily. Why? Because they have both components, the organic and the inorganic, together in one composite. So it's a great material. It bears weight, it's resilient, it doesn't shatter, it has some ability to even bend a little bit and restore its shape. Okay, but this actually presents problems in these two sets of properties. If we want to visualize bones microscopically, the way we want to do in this course, you have to do something to the bone to be able to cut it or grind it or something so that you can look into it. Okay, so remember this picture just from a few minutes ago? This is the vertebra and the cartilage. So what's been done here is that this piece of tissue has been soaked in mineral before it had, in acid to remove the mineral before it was embedded and sectioned. And then it just becomes like an ordinary piece of tissue. You can section it with a knife, a microtome, and you can compare the bone to the cartilage. Okay, but this is spongy bone, and this is spongy bone in a section at a lower magnification. You can see blood vessels throughout the bone. You can see the cells. These are called trabeculae. This is, sorry, I said this is spongy. This is compact. Okay, only when you get down to this region it might get spongy. This is compact bone. The bone in one of these trabecula and the bone in this compact region, it's the same kind of tissue. It has cells, it has matrix, it has blood vessels, it has, but it doesn't have, and it normally has mineral, but not in these preparations. The mineral has been taken away. But you can see the bone has different potential morphologies. And this is marrow here. This is marrow and blood. Okay. But remember, if we want to look at the bone with the mineral present, we have to do something else. And what do? We can take out the organic components by a solvent like phenol, and then we can saw the bone open, and we can look at the spongy bone and the compact bone. Okay, so it's equivalent to what we're looking at here, but now we're just looking at the kind of the fossils in a way, the mineral that's left behind. All of the cells are gone, and all of the organic matrix is gone, and only the mineral is present. And we can saw it open. If you try to saw open a bone without treating it like that, it would splinter and gum up the saw. It just wouldn't work. Okay, so that's a way of looking at bone, and you see aspects of the bone that you miss in the sections. Now, this is a microscopic view of a piece of compact bone. So how did we get to this point? So a saw is not good enough. It doesn't make thin enough cuts. But you can take a piece of bone and you can grind it with a grinding wheel and an abrasive surface until it's very, very thin. So you have this little solid wafer. It's not a section. It's just it's called a ground bone. Okay, and it's just like a wafer. And then you take that and you drop some ink on it, some India ink, and the India ink will fill up all these little nooks and crannies. So where the cells used to be, because they're not there anymore, gets filled up with ink. And those osteocytes would normally be there. And some of the blood vessels get filled up. Some of them don't get filled up. But you can see that there are holes where the blood vessels normally course through. And you can see some of the blood vessels are cut in cross-section. Most of them are here. And some of them are cut longitudinally. These are called haversian canals, and these are called Volkmann's canals. And I'll show you why that, how they arise in a schematic in a minute. But you can also see that the bone is layered. So some of it looks like target patterns, and some of it looks like wedges beneath these target patterns. These targets are actually cross-sections of cylinders. The cylinders are called haversian systems surrounding the haversian canals. So I just want you to kind of compare what you're looking at here to what we looked at here. So this is compact bone here. You can see the cells, you can see the blood vessels, you can see possibly a hint of the layering, but you totally miss what you're seeing here. So we have to look at it in both ways in order to understand how the bone is constructed. So these cylinders are all oriented in the same way, and they're called haversian systems in compact bone. And they're made of layers of osteocytes and the matrix that the osteocytes have produced. And the matrix is collagen and hydroxyapatite. And the collagen, from layer to layer, changes its orientation. So it's like a dense, regular connective tissue, but in layers. Remember what we saw in the cornea? We saw those layers of collagen that changed their orientation abruptly. Well, it's like that in bone as well. Okay, now if we look at these individual cells, and this is in a preparation where the mineral has been removed, and this is a very unusual optical technique, we could see that the osteocytes have projections from them. They're almost like the dendrites of a nerve cell. They're long extensions, and the cells communicate with each other and touch each other at gap junctions. And so this is how the bone cells are organized. So if we're looking here and we're looking at these bone cells, they're putting out projections. But this is a rock solid, massive mineral here. How do they do that? Well, there are little channels. As the bone develops, the cells are already contacting each other, and the mineral and the matrix is forming around them, leaving tiny little channels, which are called canaliculi, which is Latin for tiny little channels. So here is a schematic, and it's kind of cut away like a layer cake, but you can see that the target patterns are due to these cylinders. The cylinders are due to the layering. The collagen is changing its orientation at each level. The osteonal artery or haversian capillary is going through the center, and cross-cutting between these adjacent haversian canals are cross capillaries, and that's where the Volkmann's canals are, for those crossing capillaries. And the bone as a whole has an endosteum. So imagine the bone is a hollow structure. It has a marrow cavity, and the marrow cavity is lined by a layer of flattened cells called endosteum. And those cells can give rise to new bone if needed. And around the outside of the whole bone is a periosteum, which is a dense connective tissue, dense irregular connective tissue, but the inner layer of the periosteum is osteogenic. It can give rise to new bone cells. So you can get bone arising from the outer surface and from the inner surface. And you can see these canaliculi between the lacunae that the bone cells are sitting in. And we can see that better in this ground preparation. Here's a haversian canal. And here are the cells sitting in lacunae, and these rays between the different lacunae are the canaliculi, and they fill with ink in this preparation. They're very fine canals that the processes of the cell are coursing through. So going back to this, these extensions, the cell processes are going through these canaliculi. And you can see the layering here. Okay, so this is a review. If you demineralize the bone, you can section it. If you remove the organic components, you can grind it down, and you can look at different features in the different preparations. And here's a very nice three-dimensional rendition of the haversian systems, the capillaries, the haversian canals, the Volkmann's canals, the endosteum, the periosteum. These lamellae, these layers in the haversian canals are called concentric lamellae. The ones that are around the outside are called the circumferential lamellae. The ones that track the marrow cavity are called endosteal lamellae. And there are lamellae also between the cylinders, because you can't densely pack cylinders. If you take cylinders and you put them in parallel, there's always little spaces. So there are interstitial lamellae also that are not target-shaped. Okay, and this is just a piece of this showing the blood vessel and the layering and everything. Now I want to indicate that this layering of the collagen is something that is acquired through development. The bone, when it first develops, doesn't start out that way. It starts out with random collagen. And I'll be talking about this later in the term. And then it just gets remodeled over time to make the very regular patterns. There's one major part of the cranium, or the jaw, that doesn't undergo this process of layering. The alveolar bone that the teeth are embedded in remains with the collagen randomly oriented. It's a very dense bone, but it is not lamellar bone. It's not layered bone. But that's really exceptional. All the other bone in the body is layered bone. Yes?
Does endosteum surround all the canals except the haversian and the Volkmann's?
No. Apparently not. I mean, there's some indication. This remnant over here, it might be the remnant of an endothelial cell of the capillary, or it might be a bone-type cell there, but it's not the same as an endosteal layer. Okay, now there's another type of cell that's part of bone. It's very important when bone is developing, but it remains important in the adult as well, because it keeps the bone from overgrowing. If the bone fractures, it helps the bone remodel. If the bone undergoes tension because of exercise, the bone thickens in some places and thins in other places. And this population is called the Osteoclast. And it sounds like osteoblast, osteocyte. Osteo just means bone, but it's a different lineage. It comes from monocytes in the circulation. And the monocytes fuse and become cells that break down bone. They break down bone matrix. And in this section, this is a developing bone. This is the end. These are the end regions, and they haven't. There's still cartilage. Bone is forming over here. This is marrow, but over here at this interface are osteoclasts, and I'm going to zoom in. So here, this is cartilage. This is bone, and this very densely stained cell. And as we zoom in further, you'll see it has several nuclei, 4, 5, 6 nuclei, because the cells result from fusion of monocytes. And the things that the histologist or the experimenter does to prepare bone, these cells do as well. They take away the mineral, and they take away the organic components of bone matrix. So they wreck bone, and they form little canals, and they chop it away, and they're under hormonal control. And the osteoblasts of the endosteum, or the cells that are released when they get carved, when the matrix gets carved away, restore the bone. So there's a constant battle between the osteoclasts chewing away bone and the osteoblasts providing new bone. And both of them are under hormonal control. Okay, here's a higher magnification, and you can see these are osteocytes over here, but you can see that this is a very different looking kind of cell, very highly stained cytoplasm and several nuclei, and the cell is very large, and it has a ruffled border, and it is putting out acids that break down the mineral, and is putting out enzymes that chew up the organic components. And here's even a higher magnification. It's an osteoclast, another osteoclast, a very ruffled border, many nuclei, very densely, deeply stained, usually basophilic cytoplasm. And these are just ordinary bone cells here. Okay, so here is a schematic, and the light kind of purplish pink is the cell, and the cells, these are the cell's processes. Okay, this darker area is a domain that's of the matrix that is being acted on. So this is between the cell processes, and here's the bone matrix, and a microenvironment of low pH with lysosomal enzymes is being spewed out over here and breaking down the bone matrix. And these, the little spaces that are created by the osteoclasts are called Hauschip's lacunae. They're different from the lacunae that the bone cells are sitting in. They're much bigger, and they're just kind of carved out spaces. And here is a schematic of bone remodeling. So here in each panel, the bone is this light area beneath. And here is here's the marrow space here, and here are osteoclasts, committed osteoclast precursors. And these fuse, and they form osteoclasts. And the osteoclast digs deep into the they dig deep into the bone, and the osteoblasts line up here and start restoring what the osteoclasts chewed away. And when the bone gets restored, there's always a cement line. There's an indication that the repair has occurred. But these cells have done the repair. So these are osteoblasts, and these are osteocytes, and these are osteoclasts. Okay? So any questions? No? Okay, have a good weekend.
So you're a cut it. But how do you have to take out like the minerals and the organics?
Right.
So when people do like bone replacements, like hip replacements or knee replacements,
Right.
do they have to inject anything in before doing those to
Yeah.
the bones?
So, yeah, probably surgeons use very high-speed saws with diamond edges or something to cut. You know, yeah, it's just a different thing from what you'd use in histology. But you know, it's not impossible to take that material and cut it. And of course, you have to do it surgery, even if it splinters at the edge.
Yeah.
You just have to debride it. Yeah.
And then what was the bone in the jaw that you mentioned?
Oh, it's called alveolar bone. It's just the tooth sockets.
So is that why like osteonecrosis of the jaw, like I feel like that can happen with people who have osteoporosis, is that like related to that?